In this video, I'll be spending more time going over what goes on in M phase. So remember, we talked about the cell cycle, and the cell cycle started off with interphase, G1, S, and G2. Those are going to be the steps that are going to help prepare the cell in order to enter M phase, or the mitotic phase. Now, in the mitotic phase, you're going to have the 4 stages of mitosis, which is prophase, metaphase, anaphase, and telophase. And then the last step, which is not a part of mitosis, but is going to end the M phase, is going to be cytokinesis. Now you can either do this first or last. I'm going to leave this up to you. So over here on the left-hand side, if you have the notes printed out, then it's already written for you. But if you don't have a printer at home, over here, you can write the cell cycle. So we're going to briefly go over all the steps in the cell cycle, even including interphase, but just do a small little bit. We're not going to go specifically into G1, S, and G2 since we already did that in the last video. But you can draw little arrows. Like I said, you can either do that now or you can do this at the very end. So all 6 right here encompass the entire cell cycle. Now, when we get into prophase, metaphase, anaphase, and telophase, remember that is specifically going to be the 4 steps of mitosis. I like to think of mitosis as PMAT. Okay, P stands for prophase, M stands for metaphase, A for anaphase, and T for telophase. But PMAT is just a reminder of the order of those 4 steps of mitosis. All right, so now that we have that, I want to start up here with the picture of interphase. Now, when a new cell enters interphase, it's going to be quite small. And remember, interphase is going to consist of G1, S, and G2. So the cell is going to spend time growing, making more organelles, and making sure that it's ready to divide when it goes into M phase, specifically starting mitosis. Now, during this specific phase, we're going to be talking about the nucleus and the nuclear envelope here in a second, but just know the nuclear envelope is fully formed at this point, where it's fully intact. And at this point, the DNA is in chromatin form. So when we draw our little picture, we're going to make sure that we draw it in chromatin form, which means it's going to look like long, thin strings. So over here in our circle, which is our cell, that outer part is the cell membrane. And then on the inside, I'm going to go ahead and draw the nucleus. So obviously we're talking about cell division with a eukaryotic cell because eukaryotic cells are the only ones with a nucleus. So here's our nucleus. This little outer rim is going to be the nuclear envelope, just that outer edge of the nucleus. The DNA is in chromatin form, so it's going to look like these long little thin, skinny, kind of like noodles. And then the last new thing are these little things, little rectangle-looking things. These are called centrioles. And the centrioles are going to push against the cell and help get that cell broken open during cell division. And you're going to have these little spindles sticking out, and I'll label those in the next picture. But that is our cell, what it looks like in interphase. Okay, now we're going to go on to prophase. So we're going to start to get into the M phase or the mitotic phase where we start mitosis. So after G1, S, and G2, the cell is then going to enter prophase. During prophase, the nuclear envelope of the nucleus is going to disappear. It goes away. So what that means is the DNA, that chromatin, is going to be fully exposed. And actually, the chromatin is going to be turning into chromosomes. The spindle fibers, which we're going to label, are going to elongate, get longer. And the centrioles move to the outside. So I'm just going to draw a little arrow showing that we're going into prophase. I'm going to draw a dotted line to show that the envelope is disappearing. That nuclear envelope's going away. Now at this point, remember the DNA has duplicated in the S phase. So we're going to see the chromosomes actually look like little X's. So it's going to look like this. Now, in reality, there would be a lot more chromosomes going on, but I am going to draw these 4 duplicated chromosomes here. Okay, and then the centrioles are starting to move to the outside. And the spindles are elongated, they're getting longer. Now we didn't label the spindle fibers up top. I had them up there surrounding the centrioles, those little bitty lines up here, but I want you to go ahead and label them down in this because you can obviously see that they are stretching out, okay? They are elongating because they're gonna be used here in the next step. Okay, let's go on to metaphase. Now, metaphase goes after prophase. During metaphase, this step is pretty simple. Okay, prophase is actually going to be the longest one of mitosis. But during metaphase, I always like to think of metaphase and middle. They both start with the letter M. And this is because the chromosomes are going to move to the middle. In other words, the chromosomes are going to align themselves into the center. I'm going to draw another arrow. So here, the chromosomes move to the center or the middle. And at this point, you can see the centrioles are all the way on the outside. And how do they get to the middle? Well, the spindle fibers are going to attach to the center of those chromosomes. It's actually called the centromere, but we're not going to worry about that. But those fibers grab onto those chromosomes and drag them there to the center. Okay, let's go on to anaphase. Now, just like metaphase, I like to think of anaphase starts with the letter A. Anaphase, the chromosomes are going to be moved apart or pulled apart. So the chromosomes are pulled apart. Anaphase A, apart starts with A. And then the other thing that's important to note is that the cell is starting to get stretched out. So the cell membrane is actually, you can start to see it looks more oval instead of circle. Okay, I'm going to draw another arrow. And again, instead of making the cell look more circular, I'm going to make it look more oval. So you can start to see the cell, these centrioles, the cell is starting to move apart and starting to form those two cells. So again, the chromosomes, that X shape, they get ripped in half. They get pulled apart. So just do your best on the drawing. Just show them that those X's, those chromosomes, are actually apart now. And the spindles are that's what's going to be pulling those chromosomes apart, those little string-like structures. Let's go on to telophase, which is the last phase of mitosis. Now in this one, the nuclear envelope is reappearing. In other words, it is coming back. So this is the opposite of prophase, where we saw it going away. So now it's coming back or reappearing. And then the cell starts to pinch into two. And they're not fully separated at this point, but they're getting there. Draw another arrow. So again, you can start to see those two new cells starting to form. You can see that those spindle fibers are shrinking. They're no longer needed. They already did their job of pulling apart those chromosomes, half to one side, half to the other side. You can see the envelope is coming back. Notice we have an envelope in each of those two cells. And we have DNA sitting in there. Now we have to remember that prophase all the way down to telophase, that is specifically just mitosis, PMAT. The last step of the cell cycle, which is still a part of M phase but not a part of mitosis, is cytokinesis. So during cytokinesis, the two cells are fully separated. So now they are fully split. They are two new identical cells. The DNA is going to be fully back in chromatin form. So it's no longer in those chromosomes. So instead of being in those tight coils, it is now kind of stretched out back to those long, thin strings. The nuclear envelope is fully back. And those two brand new cells are going to enter interphase. And they're going to do this all over again when they're ready to divide. Draw my last arrow. I have two identical cells, two new cells. There's my centrioles. There's that nuclear envelope fully intact. There's my DNA and those long, thin strings. And again, those cells will go back through interphase and go through G1S, G2. Those organelles will copy themselves. They'll make more DNA. Those cells will get bigger. And again, will enter mitosis once again when they're ready to divide.